Good morning to everyone. It's Thursday morning, it's 10 o'clock, so it's time to have fun, enjoy science, and in fact, another wonderful clinician scientist with a very interesting topic. So it's my pleasure to say thank you and hello to Dr. Jason Hughes, and of course to say hello to all of you, because again, without you, the webinars make no sense. So thank you for choosing us. And of course, I have to say thank you very much to Emily and to James, because they are organizing and keeping this running. So thank you for all your work. To say thank you to Dr. Giulio Draeta and also to Dr. Ignacio Vistuba, because this is run in parallel by the Chief Scientific Officer Office and the TMP department. And with this, let's focus on our guest. So Dr. Jason Hughes, it's very well known in the institution and around the institution and all over the worldwide, because he is doing wonderful work at the combination of two very interesting topics, neuro-oncology and cancer biology. He's a clinician scientist with absolutely outstanding preparation, Princeton, University of Pennsylvania. Then he has done the residency and Memorial Sloan Kettering. And of course, we are happy and lucky to bring him at MD Anderson a few years ago. And I can tell you that Jason, also his main office is not in TMP, because he's a clinician and a pathologist doing clinical work. I want to say that I see him very many times here with us in the science. Why? Because he loves science, he is driven towards science, and he loves to make discovery. And in fact, a lot of grants, a lot of awards, and a lot of wonderful publications that you can see. But let's give him the light. Thank you, Jason, for being here. We are very happy to have you, and we are very curious about your presentation. Good luck.
Well, thanks, George, for the overly generous introduction. I really appreciate it, and it's really a pleasure to present at this session. So my group, we study the molecular pathogenesis of primary and secondary brain cancer. We tend to focus more on transcriptional epigenetic disease mechanisms, but we really just like to follow where the science points us. And we find that we enjoy things more that way, to use the operative word of this session. So I'm going to talk about two things today. First, I'm going to talk to you about some work we did to characterize targetable abnormalities across multiple histologies of brain metastases using an integrated molecular profiling approach. And then the second part of the talk, I'm going to go through some work that we've been doing to decipher epigenomic consequences of ATRX deficiency in malignant glioma. So yeah, let's get to it. Brain metastases are a big problem. They are the most common brain tumors, with 200,000 cases annually in the United States. They can arise even if peripheral disease is well controlled. In fact, they tend to be the final frontier of cancer progression in an individual, and a particularly deadly complication. The median survival is only 3 to 27 months following diagnosis. And sadly, most clinical trials actually exclude patients with brain met, so they tend to miss out on a lot of the other developments in science. So lots of studies have been done on this. This is one in particular I want to point out, led by Priscilla Brastianos and Bill Hahn, that brain mets are genomically heterogeneous. In particular, they can often be characterized by mutations that differ them or that differentiate them from their cognate primary lesion. And the clinical significance of these findings are underscored by the involvement in these mutations of classical oncogenic signaling networks. However, despite this notable heterogeneity and the fact that you actually have to treat brain metastases different than their primaries, at least that's what the implication of this work is, there aren't a lot of genomic abnormalities that track with brain metastases themselves. And this makes sense in a way, because you would expect the process of colonization and outgrowth in a foreign environment like the brain would be complex and require biological processes requiring the integration of multiple adaptive signals. And so, interestingly though, the PI3 kinase pathway appears to potentially be a component of this, and its upregulation has been implicated in brain metastases arising from melanoma, as well as adenocarcinoma of the breast. Okay, so interestingly enough, brain metastasis is not a universal feature of brain cancer. Some cancers metastasize frequently to the brain, like lung, breast, RCC, and melanoma. And some cancers metastasize more rarely to the brain. And so our overarching hypothesis when we embarked on this study was that a limited set of molecular mechanisms mediate the essential steps of brain colonization and metastasis. And the approach we decided to take was to assemble case-match sets that included brain metastasis, primary or extracranial metastasis, and blood or some other normal tissue from a spectrum of cancers, including breast, lung, and renal cell cancers. And we focused on frozen tissue, which did affect our numbers negatively, but allowed us to do integrated molecular profiling in the genomic, transcriptional, and proteomic space with whole exome sequencing, RNA-seq, and RPPA. We did this with breast, lung, and RCC because a colleague of ours, Mike Davies, was in the process of a similar study on melanoma, which was actually recently published. And our hope was to identify genomic and transcriptional features correlating with brain metastases within and across tumor type, and then to functionally validate these events in relevant preclinical modeling systems. Okay, so just to go through the genomic data first, this is whole exome sequencing, and it revealed the expected patterns of molecular alteration given the cancer types that we studied, breast, lung, renal cell carcinoma. And as in previous work, there was heterogeneity with regard to the actual patterns of mutations and copy number aberrations. There were differences between primary cancers and their respective brain metastases. However, also consistent with earlier work, we did not identify abnormalities specifically associated with brain metastasis. But to reemphasize, we didn't really expect to. In fact, our study was largely underpowered to do so. What we were more interested in was integrating the transcriptional and proteomic signatures to identify complex molecular pathways associated with brain mets across tumor type. And so these are multidimensional scaling plots of RNA-seq and RPPA data, respectively. These are similar to principal component analyses. It basically arranges the transcriptional profile of each sample in three-dimensional space relative to other samples as a means to identify related clusters. And what we see here is that for the most part, brain mets and primaries tend to cluster together in accordance with their tumor histology, lung, breast, or RCC. And this was reassuring to us because we were concerned that brain material in the background of these bulk transcriptional analysis would skew the transcriptional and proteomic signatures so as to obscure substantive changes occurring in the tumor cells themselves. And these data indicated, at least to us, that such effects at present were modest at best. Okay, so what did we find? All right, so the proteomic data emphatically underscored earlier findings from individual cancers that I spoke to earlier, namely that canonical oncogenic signaling networks like PI3 kinase, AKT, mTOR, and sort of the large extended signaling networks affixed to them were upregulated in brain metastasis. And there's a lot of crosstalk between these pathways with many shared components, and you can see their generalized upregulation in this heat map. What cell biology is being driven by this pathway engagement is still unclear. I'll talk a little bit more about that in a couple of slides. But we also found evidence of reduced cellular immunity in brain metastasis from our RNA-seq data. So there were reduced associations with a number of immune-related signatures, as you can see here. And here we use an algorithm called MCP counter to examine the transcriptional signatures of a variety of immune cell components. And we saw reductions in several, but with a particular focus on T cells and their variants. And we found this latter distinction to be clinically significant. In particular, patients with low T cells in their brain metastases had significantly reduced overall survival. And so we validated this finding using multiplex immunofluorescence in a partially overlapping set of formalin-fixed paraffin-embedded samples. And here's some representative image. This was multiplexed IF. Once again, we saw reduced T cells in brain mets, and further emphasizing the clinical relevance of this finding, these increased T cells in brain mets were also associated with higher levels of PD-1 and PD-L1 expression, presumably in brain met-associated lymphocytes and neoplastic cells, respectively. All right. Finally, we looked at our gene expression signatures again, and we found increased association in brain metastases were enriched in metabolic pathways, specifically oxidative phosphorylation and pathways involving the respiratory chain of mitochondria. So the transcriptional signatures are shown here. And in the vast majority of cases, the transcriptional associations with OXPHOS, which we did by the OP index or an OXPHOS score, increased from primary to extracranial metastasis to brain metastasis across the same individual. And interestingly, we found positive correlations between OP score and RPPA data for AKT and mTOR pathway signaling, suggesting that the larger PI3 kinase pathway may be in part serving to alter the metabolic profile of brain metastases to adapt to the unique nutritional microenvironment of the brain. Sorry, I had a phone call. All right, and the microenvironment of the brain is very, very unique. It includes high levels of lipids, particularly polyunsaturated fatty acids, dynamic glucose and oxygen consumption, segregated anaerobic and aerobic metabolism in astrocytes and neurons, respectively. So PI3 kinase is a complicated pathway. And so what we're doing now is investigating all of the little subnetworks of the PI3 kinase pathway and the RAS-MAP kinase pathway to determine which elements may induce OXPHOS and could potentially be targetable. Okay, but in the meantime, we wanted to ask whether brain metastasis would be selectively sensitive to OXPHOS inhibition. And so we approach this in two ways. The first, we use isogenic cell line pairs. So these are both derived from breast cancer, and in each case we have the parental line and a derivative line that's been obtained through repeated sorting through the mouse and extraction through the brain. So essentially a brain metastatic tropic derivative. And so we found in both cases, the brain metastatic tropic derivative was more sensitive to treatment with IAX10759, which is a specific OXPHOS inhibitor developed by MD Anderson. And this reduced viability was associated with increased apoptosis. And so moreover, we found that these effects were also associated with profound reductions in OXPHOS by seahorse analysis, which you can see for both cell lines here. All right. And so finally, then we moved to an in vivo model that was developed by our collaborator Bizrat Debeb's group, and it's based on an inflammatory breast cancer. And the concept here is that the cancer cells, when injected into the tail vein, form predominantly brain metastases in murine hosts. So our basic experimental paradigm is shown here. We injected the cells via the tail vein. We waited a few months, and we either treated the mice with vehicle alone or with IAX10759. And we found that with the IAX treatment, dramatically reduced brain metastases formation by luciferase imaging, but then we also did the histopathological analysis, and we're able to demonstrate vastly reduced brain metastasis number as well as overall brain metastasis cross-sectional burden. So this is very exciting to us, and it suggested that OXPHOS inhibition could be a way of actually precluding brain metastasis formation. Okay, so just to summarize the first part of the talk, brain metastases demonstrate idiosyncratic patterns of genomic evolution relative to case-matched primary tumors and extracranial metastases. Brain metastases exhibit upregulated PI3 kinase AKT mTOR signaling across tumor type. Brain metastases are characterized by clinically significant relative deficiencies in cellular immunity, and brain metastases demonstrate upregulated OXPHOS across tumor type and heightened sensitivity to OXPHOS inhibition. So, like I said, we were very intrigued by this latter finding in particular because of the potential of therapeutic options that might be available. However, unfortunately, IAX10759 was found to have significant toxicities in the early stage clinical trials. And so, as I mentioned earlier, now we're looking at other ways to perhaps target OXPHOS by targeting instead the oncogenic signaling networks that might be selectively activating it in the context of brain metastases. And we're also using a spatial profiling approach to revisit the immune phenotype that I demonstrated to you. And so hopefully I will be able to tell you more about both of those studies soon. Okay, so now I'm going to shift proverbial gears and start talking about malignant glioma. So to contrast to brain metastases, which are the most common brain tumors in general, malignant gliomas represent the most common primary brain tumors, and they are almost uniformly incurable despite their molecular and clinical heterogeneity. Some will typically lead to patient death in 15 months, others in 15 years, but they all seem to invariably lead to mortality. And this has to do with in part with the fact that they tend to widely infiltrate into surrounding brain tissue, which effectively precludes their surgical resection. And once again, despite all of the molecular and clinical heterogeneity they exhibit, they tend to be treated the same way with combinations of surgery, ionizing radiation, and cytotoxic chemotherapy. And so it would behoove us to perhaps stratify them out by their molecular features and find ways to selectively target each of the individual subtypes. And we've been given a lot of assistance in that regard over the last 10 to 15 years because malignant glioma has been subjected to extensive genomic and molecular profiling efforts. And these have delineated new patterns of molecular alterations involving genes that hadn't really previously been implicated in glioma or in other cancers. And a lot of these genes have direct influence over epigenomic landscapes. They're, in fact, epigenetic regulator genes. And I'm going to be talking about one pretty extensively today. It's ATRX. So loss of function mutations in ATRX are actually defining features of large subclasses of glioma in both adult and pediatric populations. And interestingly enough, in both adult and pediatric populations, they tend to pair with other molecular abnormalities that also dysregulate the epigenome. Specifically in adult, we see there almost invariable correlation with mutations in isocitrate dehydrogenase 1 or isocitrate dehydrogenase 2, the IDH enzymes. And in kids, we see them always co-occurring with H3.3 histone mutations. And these, like I said, both of these has established effects on the global epigenome. And so it's interesting what sort of combinatorial effects might be taking place here. I won't get into that today, but it's something that we definitely intend to head into in future work. All right. So regardless of what ATRX does, pretty much overnight, it became one of the most important clinical biomarkers we use in the histopathological diagnosis of glioma, because it so effectively stratifies one group of gliomas from another, as you can see here, and because it's so easy to assess immunohistochemically. These are loss of function mutations. And in ATRX mutant tumors, you see loss of expression in the tumor cell nuclei with retention of expression in vasculature and other normal associated structures in the CNS. And so, like I said, we literally use this every day, pretty much without knowing actually what ATRX mutation actually does. And so I'm going to talk about that for the rest of the talk. So ATRX stands for alpha thalassemia mental retardation X-linked. And it's because these same loss of function mutations in ATRX that you see in cancer also affect or are also found, can also be found in the germline. And those patients, they at least don't get cancer initially, although recent data suggests that they do develop a narrow spectrum of cancers perhaps later in life, but they're characterized primarily by intellectual disability. So as I said, these are loss of function mutations, and ATRX itself is a chromatin binding protein. And it interacts with chromatin in a variety of different ways, but perhaps its most well-described function is to partner with the histone chaperone called DAX and introduce H3.3 monomers into areas of the genome. And that way mediate chromatin compaction, accessibility, whatnot. And I said, loss of function mutations, ATRX, have been found at high rates, but in a fairly narrow spectrum of tumors, which include gliomas. And in gliomas, at least, and in other tumors as well, there seems to be this invariable association of ATRX mutations with P53 mutations. And that's likely because ATRX deficiency induces significant levels of replication stress and DNA damage, which then induce P53 dependent cell death. So from a teleological perspective, as a cancer cell, you don't want to have P53 around when you have ATRX deficiency. And we've done some work to demonstrate that this replication stress operates through G-quadruplex DNA secondary structures and in a manner that can actually be targeted therapeutically through a synthetic lethal strategy. And I won't talk anything more about that today. It's published work. If you guys want to look into it, please do. But ATRX is also fundamentally an epigenetic regulator. And it's been implicated in the regulation and the placement of a variety of different histone monomers and histone marks, along with altered gene expression. And we've waded into this pretty extensively over the last couple of years, and including with the publication here, which I'll only briefly touch on later before proceeding to primarily unpublished work. Okay, but the phenotype that ATRX deficiency is perhaps most strongly associated with of cancer relevance is a telomeric phenotype. And in particular, ATRX deficiency has been tightly associated with this phenotype called alternative lengthening of telomeres. And so the idea here is that cancer cells, like any dividing cell, they need to maintain their telomere length. And most cancers do this by activating telomerase, which is the enzyme that normally adds repeats to your telomeres. However, ATRX deficient cancers do it differently. They disable the telomerase mediated mechanism somehow and instead activate an alternative mechanism of telomere maintenance based on homologous recombination, which ends up being recognized as these ultra bright foci by telomere fish, as you can appreciate here. So this is essentially like any other telomere maintenance mechanism as a vehicle to cellular immortality. And we know it's important in gliomas because gliomas that do not have ATRX mutation, so here's the one, they're the ones that do have ATRX mutation, the primary adult subclasses that don't have ATRX mutation are instead characterized by activating mutations in the TERT promoter, which cause overexpression of TERT and pathological telomere maintenance that way. So we know it's vitally important for gliomagenesis, some form of abnormal telomere maintenance. That being said, I would argue that the same genetic evidence argues that ATRX deficiency has to be doing additional things, additional pathogenic things in addition to telomere maintenance, because there isn't a direct phenocopy here between ATRX mutation and TERT mutation. In fact, there are these other alterations that invariably occur in IDH mutant gliomas that don't have ATRX deficiency, in particular this large deletion of chromosomes 1p and 19q. So the phenotype isn't complete. And furthermore, while adults have very high rates of TERT mutation in gliomas that don't have ATRX mutations, you don't see the same thing in kids. In fact, very few pediatric gliomas have TERT mutations. So once again, this argues that ATRX induces an epigenomic dysfunction that itself contributes to gliomagenesis. And we wanted to find out what that was. So we designed a variety of different experimental model systems to do this. And the one we got the most leverage on so far has actually been a murine system. It's based on ex vivo murine neuroepithelial progenitor cells that we culture directly from the brains of pups. And neuroepithelial progenitor cells are thought by many to essentially effectively model cells of origin for human glioma. And so by using p53 deficient and ATRX deficient mice, crossing them and extracting various cell lines, we were able to essentially generate isogenic pairs of neuroepithelial progenitor cells with either p53 deficiency alone or p53 deficiency with ATRX deficiency. And we noticed a phenotypic shift in the ATRX deficient cells right away. Where they lost their conventional sort of bald floating structure and instead spread out in a monolayer at the bottom of the plate. So we were intrigued by this, and there were actually pretty profound transcriptional distinctions between these two groups. But what did not occur is that these tumors did not form a tumor any more readily and did not proliferate appreciably faster. And while this was initially disappointing to us, we ultimately felt that this made sense because ATRX deficient tumors, particularly in the adult, actually arise very slowly and indolate for long periods of time. And so we were more interested in investigating what other disease-relevant phenotypes might be emerging through the deletion of ATRX in this context. And we looked at the transcriptional data and we found increased associations with developmental and cell motility phenotypes primarily. So to dig into that a little bit more, we did a differentiation assay. Shown here, we actually essentially just differentiated the cells in culture over a period of a few days. And what we found when we looked at key marker expression was that the normal markers of neuronal differentiation here, the ATRX deficient tumors here are shown in the solid lines. The normal markers of neuronal and oligodendroglial differentiation were abrogated in favor of increased GFAP expression, sort of a canonical astrocytic marker. Although I need to remind you that GFAP also is a pretty avid marker of neuronal stem cells. So essentially, how we interpret this data is it just abrogates the normal neuronal differentiation profile of neuroepithelial progenitors. And at the same time, using a couple of different assays, we saw increased motility in ATRX deficient cells. And so we thought this was very interesting. And by just eliminating one gene in a p53 deficient context, we were able to recapitulate two of the cardinal phenotypes that are associated with the diseases in question. That is diffuse infiltration and a nominally astrocytic differentiation profile. So we wanted to see exactly how we were doing this. So the first thing we did was we wanted to figure out where ATRX normally lived in the genome. And so we did ChIP-seq, and this took some time. It's really not an easy thing to do. But the results were actually quite informative. We did it in our neuroepithelial progenitor cells. And the only previous data that had been gathered on ATRX localization by ChIP-seq was in embryonic stem cells. And what that showed was a primarily intragenic binding pattern. But we found something completely different in neuroepithelial progenitor cells. It was much more gene associated and much more, it looked much more like a transcriptional regulatory signature. And this may actually speak to a fairly defined cell type specificity associated with ATRX deficiency. And it makes sense given the fairly narrow spectrum of tumors with high rates of ATRX deficiency. Okay. And so we wanted to get a sense of what this was doing, what was happening at these binding sites and at other sites in the genome in response to ATRX deficiency. And so we did a TAC-seq, looking at chromatin accessibility in both the ATRX intact and ATRX deficient context. And then we were able to ascertain areas of the genome that either had increased opening or increased closing in response to ATRX deficiency. We call them open and closed regions. And what was surprising is that the vast majority of them had direct positional overlap with where an ATRX binding peak would normally be. To suggest that the effects on chromatin accessibility are primarily restricted to the immediate vicinity of normal ATRX binding peaks, in other words, vacant ATRX binding sites. And then we explored genes that were regulated by this process. They were involved in cell motility and differentiation. We also demonstrated that there was misincorporation of histone 3 at these sites as well. And this led us to our first model for what was going on here, which we've since developed, as I'll show you later, which is that ATRX, when it's lost, the areas in the immediate vicinity of ATRX binding sites become dysregulated in terms of their accessibility, in part through misincorporation of histone 3 monomers. And this leads to differences in gene expression, which then impact disease-defining cellular phenotypes. All right, so we got a sense that this might just be the tip of the iceberg, and we wanted to get a more complete idea of what was going on in terms of chromatin regulation, chromatin states, chromatin architecture. And so in collaboration with Kunal Rai, we embarked on a profiling exercise looking at a variety of different chromatin marks in our ATRX intact and deficient neuroepithelial progenitors. And the largest differences we saw involved repressive marks. And I'm showing you the results here for H3K9 trimethylation, which is one of the primary repressive chromatin marks and tends to be involved in both facultative and constitutive heterochromatin. And we saw here differences in peak assignments, but that was really just the tip of the iceberg. When we actually started looking across the genome, some very dramatic effects were seen. And they primarily tended to affect these regions of the genome that had large swaths of H3K9 trimethyl mark. And these regions of the genome in the setting of ATRX deficiency showed dramatically reduced H3K9 trimethylation. And at the same time, when we looked at whole genome bisulfite sequencing, which is normally well demarcated around these regions, we saw that this regulatory insulation was lost. What was interesting about these regions is they positionally overlapped with so-called laminin-associated domains, or LADs. And so it turns out that as a cell is going through its development and differentiation, genetic programs that it doesn't really need anymore are sort of locked down by H3K9 trimethylation and then essentially marginalized to the side of the nucleus. They're tacked up on the nuclear lamina, sort of like a child when you tell them to clean up its room and he or she just sort of pushes all their toys to the side to get them out of the way. And that's how these genes end up being regulated. And you can actually see the effects of laminin association of H3K9 trimethylation by immunofluorescence in ATRX intact and deficient cell lines. This membranous distribution that you see as an element of the staining pattern is lost in ATRX deficiency, and you can quantify that down here. So it turns out that LADs, or LAD misregulation, has actually been implicated in cancer. In particular, Andy Feinberg and his group at Hopkins have shown that cancers stochastically dysregulate these regions, and in the process, presumably activate latent developmental programs, which then could potentially contribute to oncogenesis. And so we looked at actually the genes that are normally associated with LADs and not associated with LADs across differentiation axes. We found that as you differentiate cells from neuroepithelial progenitors to astrocytes, the genes that have increased association with LADs normally are actually downregulated in their expression in ATRX deficiency, and the genes that normally decrease in their association are actually upregulated, suggesting that we've actually mobilized this differentiation axis with our LAD disruptions here. But we actually thought, we were thinking deeper, because that this might once again just reflect a larger underlying process going on. In particular, we thought about the implications that this might have on chromatin topology. So it turns out that your genome, whether it's laminin associated or not, tends to be localized in these self-associated regions called topology associated domains, or TADs. And these can actually be delineated by high C as these co-localization or connection plots, where these triangles actually demonstrate the individual topology associated domains. So we were wondering, does ATRX deficiency actually disrupt chromatin topology? And so we've actually done high C now on our ATRX positive and ATRX deficient NPCs, and we see some very intriguing things, although we're still working through this data. But we do see disruptions in these topology associated domains. Here's one example. As you can see, this singular domain here appears to be broken up into multiple mini TADs, or I guess you could call them neo-TADs. And this is occurring actually in one of these large regions of heterochromatin that I've been speaking to you before up until now. And so this suggests that ATRX deficiency actually fundamentally impacts chromatin topology in mediating whatever effects it has on chromatin. Okay, and in some ways this is not surprising, because ATRX has actually been shown already by others to interact productively with CTCF. So CTCF is a protein that has an established role in setting up these insulated regions of chromatin. If you look at the ATRX binding sites from our ChIP-seq data, we find that the motifs are highly enriched in CTCF binding sites, and we've actually confirmed this by ChIP-seq or by cut and tag analysis for CTCF, which I'll show you in a couple slides. But first, I want to introduce one additional concept, and this has to do with endogenous retroviruses. And so endogenous retroviral elements actually compose a significant portion of our genome. They've just accumulated over time through repeated cycles of evolution. And these include a variety of different variants, line elements, sign elements, what have you. And ATRX has actually been repeatedly implicated in the silencing of endogenous retroviral elements. And it does this likely through coordination with the CAP1, ESET, or SETDB1 complex. And SETDB1 is a methyltransferase, which adds the trimethyl marks to H3K9 moieties. Okay, so we looked at our data, and we found that, in fact, there was deregulation of ERV expression in our ATRX deficient NPCs. And in particular, this involved upregulation of L1 elements here and downregulation of so-called B1 elements or sign elements here. And this made sense because L1 elements are primarily localized to compartment B chromatin, or essentially heterochromatin LAD regions. And B1 rich regions are in the non-LAD regions. And you can actually appreciate that in this earlier work by this very elegant staining that was done. All right, so furthermore, when we used publicly available ChIP-seq data for L1. So ChIP-seq is a way to actually map the localization, or the regulatory localization of non-coding RNAs, like transcribed ERVs, and they tend not to diffuse very far from where they're initially transcribed. So it's a way to see where they're actually productively interacting with proteins and other RNAs, and also where they tend to be produced. And so we confirm here with our L1 ChIP-seq, or the ChIP-seq, that these RNAs are actually being produced in these LAD regions. And furthermore, these are the regions where we have dysregulated H3K9 trimethylation with ATRX deficiency. And we also have our ChIP-seq or our CTCF cut and tag up here, which shows a host of additional binding peaks that otherwise are not there in the ATRX intact context, suggesting that all of this dysregulation in these regions is ultimately having impacts on CTCF, K9 trimethyl, as well as endogenous retrovirus expression. Okay. And finally, if you look at the genes that are actually impacted as targets of these L1 ERVs by ChIRP-seq, you see they're highly enriched in normal neurodevelopmental programs. This is both in ATRX deficient neuroepithelial progenitors, but also in ATRX deficient gliomas using TCGA data. So this is additional compelling evidence suggests that ATRX deficiency fundamentally dysregulates normal neuronal development as a way to promote oncogenesis in these tumors. So this is all very, very complex, convoluted biology. How would you go about actually targeting this therapeutically? And so we've put our heads together on that. And one of the strategies that we came up with is to actually use the signature of ATRX deficient transcription in neuroepithelial progenitor cells as essentially a way to fish out candidates, compound candidates, and strategies that we could use to target it. So our first approach was in silico. We use the connectivity map at the Broad Institute, which is actually essentially just a compendium of transcriptional signatures from cell lines associated with different compound treatments. And we look for transcriptional signatures that essentially represented the reverse of what we were seeing with ATRX deficiency. And we found that those signatures tended to be highly enriched in HDAC inhibitors or histone deacetylase inhibitors. And so there are a lot of HDACs and there are a lot of HDAC inhibitors. So which one should we actually be targeting? So we were intrigued in particular by this one class 3 HDAC called SIRT2, because in our data, it had increased RNA-seq expression with ATRX deficiency. It was also associated with an open region with ATRX deficiency as determined by our ATAC-seq, and it had an ATRX binding site associated with it. And so we thought this was a reasonably good candidate to shoot for. We also found that it had increased expression in ATRX deficient patient-derived glioma stem cells relative to wild-type counterparts. And when we looked at two public compendiums of data from TCGA and CCGA, we found that ATRX deficient gliomas tended to have upregulated SIRT2 expression relative to ATRX wild-type glioma subclasses. So we looked more closely at SIRT2 expression using single-cell transcriptomic data, and we found that its expression was heterogeneous across our cell type. This is your typical UMAP plot for single-cell data. But the heterogeneity of SIRT2 expression tended to correlate quite well with key genes mediating ATRX deficient phenotypes like ROC, which is a gene that's involved in the migratory phenotype I showed. There were others that were involved in differentiation that I'm not showing here as well. So this raised the possibility to us that perhaps SIRT2 expression itself was a primary driver of the oncogenic and malignant phenotypes that ATRX deficiency was mediating. So we looked back at CCGA data where the survival data is actually quite mature, and we were able to demonstrate that in the IDH mutant non-codel gliomas, which are essentially the ATRX deficient gliomas, high SIRT2 expression was associated with significantly worse survival or significantly reduced survival relative to low SIRT2 expressors. And this was a correlation that was not seen in the ATRX intact subtypes. And so we were excited about potentially targeting this therapeutically. And it turns out there are multiple SIRT2 inhibitors that are available, including one, AK7, that actually crosses the blood-brain barrier. So we started doing experiments with these. We first wanted to document that, in fact, these are HDAC inhibitors. You just expect increased histone acetylation in response, and we do see that particularly in our ATRX deficient NPCs. We also, when we look at the actual signature associated with ATRX deficiency on a transcriptional level, we're able to see that these compounds are actually able to reverse that signature. And so what are the effects on the actual phenotypes of the cell? So I'll tell you right off, this treatment did not induce higher levels of cell death, and particularly at physiologically relevant concentrations. But when we actually looked at the phenotypes that we are typically associated with ATRX deficiency, we did see significant effects. So this is cell migration, and we did this in both NPCs and in patient-derived glioma stem cells, we were able to show that treatment with SIRT2 inhibition actually reverses or reverts cell migration potential essentially to what you see at baseline levels in the ATRX intact context. And this is for both human and murine contexts. Furthermore, when we looked at differentiation phenotypes, we found that treatment with SIRT2 inhibition significantly increased beta-galactosidase as a marker of cellular senescence, and did so with a good amount of cell activity, once again, in the ATRX deficient context. So there are a couple of different mechanisms that we thought could be at play here involving histone acetylation. So the most obvious one was H3K27 acetylation. This is an acetylation event typically associated with active superenhancers. It tends to counteract chromatin compaction as well. And so we confirmed actually that treatment with HEK2, AK7, and thio, our SIRT2 inhibitors, actually reverted chromatin compaction that we saw in our ATRX deficient NPCs. I apologize for the resolution here, but it increased the intensity of H3K27 acetylation, decreased the intensity of H3K27 methylation, the cognate repressive mark. And these genes that were activated tended to be associated with the negative regulation of cellular motility, for instance. So this is one potential mechanism that could be at play here. But there was something else that we looked into also, and that has to do with H4K16 acetylation. So this is another mark that tends to tag active genes and enhancers, but doesn't actually, it doesn't appear to be involved in chromatin compaction. And so when we did ChIP-seq for this mark, we were able to demonstrate increased peaks arose with SIRT2 inhibition relative to vehicle treatment, particularly in the ATRX deficient context. And interestingly, when we actually did motif analysis on these peaks, we saw that they were highly enriched in targets of this transcription factor, KLF16, and that these KLF16 targets were repressed, presumably because this H4K16 acetylation helps to recruit any KLF416, which then causes their repression. Furthermore, that these genes themselves have been implicated in senescent relevant processes. So we're excited about this, and that we may have a strategy to induce essentially terminal differentiation in ATRX deficient glioma. And now we're in the process of doing in vivo analyses using human GSCs and mice, and hopefully we'll have that data soon and be able to finish up the story. All right, so just to summarize the second part of the talk, ATRX deficiency alters chromatin accessibility and gene expression profiles with downstream effects on disease-relevant cellular phenotypes. ATRX deficiency disrupts heterochromatin integrity and chromatin topology, impacting developmental programs. And finally, SIRT2 inhibition reverts ATRX deficient phenotypes and transcriptional programs by targeting multiple histone acetylation events. All right, so that's all I had planned to talk to you about today. I just want to briefly acknowledge the people that did all of this wonderful work. So the first part of the talk was the brain metastasis work was led by Kazu Fukumura, postdoc in the lab, with computational assistance from Prit Beni Malgolwar, another postdoc in the lab. And the second part of the talk was the ATRX work was led by Prit, following up on earlier work by Carlo Danussi. And of course, we've had wonderful collaborators. Particularly, I want to acknowledge those in Kunal Rai's group for helping us with the epigenetic programming. So thanks so much for your attention. I'd be happy to take questions.
Thank you very much, Jason. Wonderful talk, and in fact, very interesting and a lot of interesting questions. So let's start. Thank you for the amazing talk. I want to ask if you tried agents targeting the DNA response pathway on ATRX deficient tumors, such as PARP, ATR inhibitors, XPO inhibitors.
Yeah, so we actually have been trying to do that, but and that has to do with the story I didn't tell you. So we've looked into DNA damage associated with ATRX deficiency. It is replication stress induced, and it appears to revolve around these weird DNA structures called G4s, G quadruplex DNA, and so. And they're essentially knots that form in your DNA, and they can lead to replication stress and DNA damage. And it turns out if you actually treat these cells with agents that stabilize the G4s and prevent them from resolving, you can dramatically increase DNA damage even further in the ATRX deficient context, sort of like a selective synthetic lethal strategy. And so we're following up on that analysis right now in bona fide preclinical models. We also have been looking a little bit at combining that therapy with CHEK1, CHEK2 inhibition, what have you, because that may work. And actually, in work that I was involved in by Tim Chan, PARP inhibition has actually been shown to be effective in IDH mutant gliomas, and those gliomas tend to be enriched in ATRX deficiency as well. So yes, these are all strategies that I think are highly worth looking into, especially since the epigenetic consequences of ATRX deficiency seem so complex and hard to target. It might be better just to slam it over the head and go after this genomic instability target instead.
Very interesting. So another question. So thank you so much for this very informative and comprehensive seminar. The question is, H3K9M3 images show heterogeneous expression on both images, ATRX negative and positive. How did you ensure unbiased analysis of these images?
Yeah, so I think we just made sure to do a lot of sequencing and to make sure that the signals we were seeing is real. I think also we tended to normalize our analysis based on the highest peaks that we see in both sessions. And so if everything was increased in its intensity, we tended to blow it off. But if there were certain regions of the genome, like these LAD regions, that had dramatically reduced signal intensity relative to other areas of the genome where it was relatively maintained, we tended to focus our analysis there. But I agree, the analysis itself can be tricky because particularly these large heterochromin domains, they don't lend themselves to facile study with your conventional peak calling algorithms because they're so broad in their distribution. And so you almost have to just kind of surf through the genome and then find other ways to quantitatively assess them.
Thank you. And another question. How does ATRX deficiency align with DNA methylations? Implication with interaction metabolism?
Yeah, so we looked at whole genome bisulfite sequencing. I'm particularly focusing on, once again, those LAD regions. And it's interesting. There doesn't seem to be a defined effect sort of plus or minus. It's more the disruptions that we see in whole genome bisulfite sequencing are more having to do with typically insulated regions of the genome, areas that typically have low levels, have increased levels. Areas that are typically have very high levels, those levels can come down a little bit. We just see basically a disruption of the integrity of the genome and sort of walling off different portions of itself from each other. And all of these signals tend to be kind of associated.
Thank you. And then a question from my side. So when you have done all these genome-wide studies, did you see some preferences for some peculiar non-coding regions, introns, other type of intergenic regions that could make sense?
Well, yeah, no, I think, like I said, these LAD regions are actually, as you know, relatively gene poor, and they're highly enriched in these retrotransposon ERV elements, right? And so that in itself, we think is very interesting and actually speaks to the non-coding genome.
Did you check for microRNAs on long non-coding RNAs in these regions?
No, we haven't specifically done that yet, but yeah, you're right. That would be very interesting to do.
Thank you. So thank you very much, Jason. Dr. Hughes, it was a wonderful talk, very interesting, and thank you to all of you who attend. And in fact, enjoy science and let's make discoveries. Thank you. And in 2 weeks from now, another wonderful presentation, this time on non-coding RNAs in metabolic disorders. Thank you again, Jason. Wonderful talk. And thank you all of you who were with us today. Good luck with everything.